Good day, everyone. This lecture video is all about the chromosomal basis of heredity. Just a quick recall, heredity is the transmission of genes or traits from parent to offspring or from one generation to the next. So under this topic, we have 4 subtopics. We will be having a quick review of the cell and its parts rather. The chromosome structure, we will study that. We will be also discussing the cell cycle and the stages of the cell division. And finally, we will be having a quick review or a quick discussion of life cycles. So to start with, we have the cell. Okay, we have here all forms of life except viruses consist of cells. Meaning to say, viruses are not considered as living organisms because the cell is the basic unit of life. So viruses are not living organisms because they require a host in order to live, all right? And the organization of each cell is the same in all organisms, whether that be a plant, an animal, or a microbe. So the cell has 3 main parts. We have the cell membrane, the cytoplasm, and the nucleus. So the cell membrane, as you all know, is also recognized or also considered as the plasma membrane. So the cell membrane is a phospholipid bilayer, and with that, it is a semi-permeable membrane, meaning to say not all substances can enter or pass through the membrane. Okay, cannot enter the inside of the cell. So the cell membrane serves as a protection or as a barrier, and that separates the inside of the cell with the external environment. All right, and then next is the cytoplasm. The cytoplasm consists of the jelly-like cytosol, and this cytosol is the fluid, the name of the fluid present in the cytoplasm. And the cytoplasm is where the organelles such as ribosomes, mitochondria, endoplasmic reticulum, Golgi body or Golgi complex, etc. are suspended in. Okay? And then finally, at the center or inside of the cell, okay, the innermost part of the cell, we have the nucleus. And the nucleus is considered as the cell's control center, or it's considered as the brain of the cell because it controls or directs the activities of the cells. Basically, the nucleus instructs the cell on what to do, right? Because the nucleus has the chromosomes, and the chromosomes are the carriers rather of hereditary materials, and the hereditary material or the genetic material contains the information or the messages that will be transcripted or translated at the end of translation process. All right, so that is the 3 main parts of the cell: cell membrane, cytoplasm, and nucleus. So we have here some pictures rather. Some pictures we have, this is actually an animal cell. At first glance, this is, you can consider this or you can tell that this is an animal cell because of its shape, okay? Normally or usually, plant cells, plant cells are more rigid in form. Say they have a structure called the cell wall, which creates or which forms the rigid structure or rigid form of their cells. So this is an animal cell, more of a rounded shape, and a plant cell, more of a rectangular shape. Okay? Next. We have here a eukaryotic cell. When we say eukaryotic cell, this is the type of cell present in eukaryotes or multicellular organisms. And we have the prokaryotic cell, which is the type of cell present in prokaryotes. For the eukaryotic cell examples, we have plant cells, animal cells, and these cells contain a nuclear membrane or yung tinatawag nating nuclear envelope. So, yun yung difference ng eukaryotic cell at saka prokaryotic cell. In the prokaryotic cell, you cannot see a nuclear envelope or a nuclear membrane. The function of the nuclear membrane is it separates the genetic material from the cytoplasm. Recall, that I previously mentioned that the genetic material is in the nucleus, correct? And the nucleus is enveloped or surrounded by a nuclear membrane, especially in the eukaryotic cell. The membrane, the nuclear envelope or the nuclear membrane, protects the genetic material inside the nucleus. And yun yung wala sa ating prokaryotic cell. So, yung eukaryotic cells, that is the type of cell present in majority of living and multicellular organisms. Prokaryotic cells, these are present in bacteria and blue-green algae. So this is the type of cell for prokaryotes, right? So instead of having a nucleus and a nuclear membrane, what is present in a prokaryotic cell is the nucleoid region. So this region of the cell, of the prokaryotic cell, already contains the chromosome, the ribosome, and of course, the fluid present here inside the nucleoid region, right? So that is the prokaryotic cell. There is no nuclear membrane, all right? Regardless whether that is a plant cell, animal cell, eukaryotic cell, or prokaryotic cell, all cells share 4 key components. We have the plasma membrane, or what we call the cell membrane, cytoplasm, where the organelles are found, DNA, the genetic material, which contains the information or messages that will be translated, and ribosomes. So ribosomes, they serve as site of photosynthesis. RNA synthesis or protein synthesis. Okay, so let's discuss yung tinatawag nating nucleus. So this is actually the nucleus, all right? The nucleus is surrounded by this nuclear envelope or nuclear membrane. And this nuclear membrane is in active contact with this endoplasmic reticulum as well as the cell membrane, right? So kung babalikan natin ang structure ng cell, lakihan natin. This is actually the nucleus. This one is the nucleus. Ito yung blue, yan yung tinatawag nating nuclear envelope or nuclear membrane connected or in active contact with the endoplasmic reticulum. This is the endoplasmic reticulum. And this endoplasmic reticulum, sabi kanina sa slide, ay connected din sa ating cell membrane. Okay? Cell membrane. Yan yung ating nucleus. All right. The nuclear content or the contents of the nucleus consists of a dark network called chromatin. This one is a dark network. So that's your chromatin. Chromatin yung tawag natin kapag hindi pa siya nag-undergo ng cell division. But once it enters the stages of cell division, we now call it as chromosomes. So that's the difference, right? And then we have here one or more spherical bodies called the nucleoli may be found attached to specific chromosome regions. We will learn a little later the different parts of the chromosomes, and one part of the chromosome is called the NOR or nucleolus organizer region. That is the region where the nucleolus forms or attaches to. Okay? So yun yung tinatawag nating nucleus, the nucleus. Speaking of the chromosome or the structure of the chromosome, the chromosome usually maintains constant shape and size at specific stages of the cell cycle. The condensed chromosome may be as short as 1/4 micron or as long as 30 microns in Gillium species. And pag sinabi natin gross structure, that's actually the structure of the chromosome as it enters the stages of cell division. So sinabi dito, mitotic chromosome. Okay? Because mitosis is a type of cell division. Okay, so gross structure, yun yung structure ng chromosome once it enters already cell division stages. Okay, so it is a rod-like body with the following parts: the centromere, or we otherwise call it as primary constrictions. Next is the secondary constriction and the nucleolus organizer or organizing region. Ito yung sinabi ko kanina. And then lastly, we have chromomeres and knobs. For the centromere, ito yung tinatawag nating centromere, located at the center. Okay? Kaya siya tinawag na centromere. It's not actually yung pinaka-center na part, like connecting equal lengths, but tinawag siyang centromere because it's the act or it's the point of association between the chromosome and the spindle. During cell division kasi, the microtubules arising from the poles, okay, from the poles at the spindle attached to the centromere. So yan yung point of attachment nila because it's in the centromere where the kinetochore proteins are present or attached to. So yun yung tinatawag nating centromere. Another is that the centromere connects the short and the long arm. So aside from the four mentioned parts kanina, we have the centromere or the primary constriction. Second is the secondary constriction. We also have the NOR or the nucleolus organizing region or organizer region. We also have the chromomeres and knobs. And apart from that, we have the P arm and the Q arm. The P arm, this one, yan yung tinatawag nating short arm. And the Q arm is the long arm. So although hindi naka-indicate dito, but this one is the P arm or the short arm, and this one is the Q arm or the long arm. So the centromere serves as the point of association. That is what connects these two arms or two segments. And another, we also call this one, this is a chromatid. This is also a chromatid. Okay. Next, this is the point at which force is exerted in the separation of dividing chromosomes. Once we arrive at anaphase, once the cell is in the anaphase stage, whether that's mitosis or meiosis, this is where the force is exerted. Kasi dito nakakabit si microtubule or tinatawag nating spindle fiber. Okay? Next, we actually have different types of chromosome based on centromere position. Ito yung sinasabi ko kanina na centromere, center, but it doesn't actually mean na doon siya sa pinaka-sentro na part makikita. Because we have here different types of chromosomes. We call it telocentric kasi yung telo, ibig sabihin nun is end or terminal part. So ibig sabihin, nasa end portion ng chromosome makikita yung kanyang centromere. Yan yung tinatawag nating telocentric. But in humans, we do not have this type of chromosome. Another is the acrocentric. Pag sinabi nating acrocentric, it's near the end. The centromere is near the end of the chromosome. So yan yung acrocentric. Acro meaning near. Okay? Next, we have submetacentric. Or acrocentric also mean away from the center. So it's near the end or away from the center. Okay? Submetacentric, pag namang submetacentric, malapit sa gitna. The centromere is near the middle. Right? Next, we have the metacentric. Ito na yung sinasabi ko kanina na both arms have the same length, equal lengths. Okay? Because the centromere is located at the center or at the middle. Yan yung tinatawag ko kanina, P arm or short arm and the Q arm and long arm. Next, we proceed to secondary constriction. Pag sinabi natin constriction, it's actually a narrow region, right? So it's a narrow region of the chromosome found at any point except doon sa centromere. Yan yung secondary constriction. So it's the pinching off of a small chromosomal section forming a satellite. So ito yung tinatawag nating satellite. So we have here a secondary constriction. We have another here, secondary constriction. May nakalagay dito na secondary constriction or nucleolar organizer region because a nucleolus organizer region or nucleolar organizer region is actually a type of secondary constriction because the constriction is located other than the position of the centromere. Okay? So ito na yung tinatawag nating NOR, nucleolus organizer region. It serves as a landmark. Okay? Landmark where there are tandemly repeated sequences of rRNA or ribosomal RNA. Okay? Dito, kaya sa tinawag na NOR because it is where the nucleolus is attached to. So ito na yung tinatawag nating nucleolus. Diyan naka-attach or dyan na-form yung nucleolus. We usually see that when we observe the chromosomes under the microscope. So ito yung tinatawag ko kanina. We have here the chromosome arm. This is the constriction or the NOR, and this is the satellite that is formed. Hindi ito yung centromere. The centromere is located here, this part. Okay? The centromere is located here. Hindi ito yung centromere because again, NOR is part or it's a type of secondary constriction. So the centromere is located in a different region. So this is the NOR consisting of these tandemly repeated rRNA genes. Next, chromomeres and knobs. These are the beads representing the chromomeres and knobs. So it says here, when a mitotic chromosome is stretched out, if you relax the chromosome, it would be observed to consist of a string of particles, characteristic particles of different sizes with different distances. So, unequal yung distances between these particles. And yung mga mapapansin natin na smaller beads, we call them chromomeres, and the larger beads are called as knobs. They actually serve as cytological markers. Next, we proceed to our third topic under this chapter, cell division. So, cell division is actually the manner wherein one parent cell is divided into two daughter cells. So, the division of one cell will produce two daughter cells. Okay? So, why do cells divide? Cells divide to give rise to new cells. So, meaning to say, cells divide to multiply. Okay? So, cells divide to multiply. So, we have, at the start of the cell division process, we have one cell. At the end of the cell division process, we produce two cells. Alright? So, yung cell division natin is we have two types, mitosis and meiosis. So, yung cell division stages actually occur after the interphase. So, yung interphase na tinatawag natin is a part of the cell cycle. So, the cell cycle consists of the interphase and the different cell division stages, yung mitosis at saka meiosis. So, bale, mas malawak yung coverage ng cell cycle than that of the cell division. Because ito lang yung part ng cell division. The cell cycle is the whole of this, right? So, it's an ordered series of events that takes place in a cell as it grows and divides. Meaning to say, nauuna yung interphase. Nauuna yung G1 kaysa S-phase. So, sunod-sunod. It's an ordered series of events. In a cell cycle, we have the interphase. Gaya ng sinabi ko kanina, interphase and the M phase. This one is the M phase. M phase refers to either mitosis phase or meiosis phase. Right? So, we have the interphase. The interphase consists of the G1, the S phase, and the G2 phase. For the G1, this is actually considered as the longest phase. So, tawag nito, dito in-spend ng cell karamihan ng time niya. Okay, so G1 phase occurs at about 8 hours or more. So what happens during the G1 phase? Yung G1 phase is also known as first gap phase or first gap period under interphase. So what happens during G1 is the cell grows, nami-meet yung normal cell function. So with cell growth, we actually mean yung mga ribosomes, mitochondria, yung mga organelles present in the cell fully form or fully develop. Okay? They duplicate. Organelle duplication, normal cell function, and the cell grows by imbibing water and nutrients doon sa cell. Okay? So after finishing G1, hindi siya pupunta agad sa S-phase. Mangyayari is i-check muna ng boundary. So, meron siyang boundary dito, and boundary dyan. So, we have here a checkpoint. We call this G1 checkpoint. The G1 checkpoint functions by checking if the cell already satisfies or meets these requirements. Nag-grow ba yung cell? Normal ba yung cell function? Na-duplicate ba yung mga organelles, etc. Kung na-meet yun, magpo-proceed siya sa S phase, wherein DNA replication occurs. So S phase, tinawag siyang S phase because it refers to synthesis. So DNA synthesis or DNA replication. And once the DNA is replicated, i-check dito sa G2 checkpoint kung na-meet yung requirements. Kung na-replicate ba yung DNA. If yes, then proceed siya sa G2 phase for protein synthesis. Alright? And kapag natapos na yung protein synthesis sa G2 phase, which lasts for about 2 to 5 hours, papasok na siya sa prophase, metaphase, anaphase, and telophase. Again, it's an ordered series of events. All of these are called mitosis. So usually it occurs 1 to 3 hours, right? So ang unang stage ng mitosis, we have the prophase, followed by metaphase. Hindi maaring prophase, then i-skip niya yung metaphase, tapos pupunta siya agad sa anaphase. No, so it's an ordered series of events, right? So speaking of mitosis, it's actually a process or a stage of cell division that wherein the genetic and chromosome composition of a cell is faithfully reproduced in each of the daughter cells. Meaning to say, at the start of mitosis, you have this number of chromosomes, and then at the end of mitosis, you produce the same number. Each cell contains the same number of chromosomes present in the parent cell. Okay. This process, mitosis, serves as a means of reproduction, especially in unicellular organisms. But in us, multicellular organisms, plants, animals, our cells undergo mitosis for growth and for repair or replacement of damaged tissues. So yun yung mitosis. For the stages, we have here prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. But the stages of mitosis, we only have 4. Yung pinakita ko kanina, we actually abbreviate it as PMAT. P, prophase, M, metaphase, A, anaphase, T, telophase, PMAT. Yung mitosis is sometimes referred to or considered as somatic cell division because this process, mitosis, occurs in somatic cells or body cells. Okay? Yung meiosis naman, it occurs or happens in our reproductive cells, in our sex cells, or in our germ cells. Okay? So yan yung mitosis, somatic cell division. So in prophase, so upon entry dito sa prophase stage, by the way, dito sa interphase stage, ang tawag pa lang natin, kung naalala ninyo yung sinabi ko kanina when we discussed the nucleus, chromatin ang tawag natin sa strands or thread-like bodies na nakikita natin sa nucleus. Chromatin pa lang kapag nasa interphase stage. But once it enters the prophase, we now call them chromosomes. Chromosomes because condensation already happens. Right? So, kailangan mag-condense ng chromosome. Pag sinabing nag-condense, ganito na yung itsura. Hindi na siya thin strands, kumbaga. Okay. Yung chromosomes present at prophase has 2 chromatids. Okay? Yun yung tinatawag nating chromatids, yung sinabi ko kanina. And then, we have here an intact nuclear envelope, but the end of prophase, this nuclear envelope should already have disintegrated, started to disintegrate, right? And then we have 2 poles here containing the centrosomes. They move towards opposite poles. And we have here the spindle. This is a whole spindle containing yung spindle fibers or what we call the microtubule. Itong spindle fibers or microtubules, dito mag-a-attach yung ating mga chromosomes. That's why kailangang mag-disintegrate or ma-break itong nuclear membrane or nuclear envelope para makalabas sila doon sa nucleus. Maka-attach sila dito sa microtubule or spindle fiber. So, pagkatapos dito sa prophase, proceed sa metaphase. Okay? Metaphase na dapat. Pero pinakita lang dito sa prometaphase na naka-arrange na nandun na sa opposite poles. Exactly at the opposite poles itong ating centrosomes. centrosomes or tinatawag nating pole, spindle pole, kasi they are located on opposite poles. This is where the spindle or the microtubules emerge from. And then, unti-unting na-attach yung centromere doon sa microtubule. Na-attach siya doon sa centromere because this is where the kinetochores are attached to. Right? And then at metaphase, finally, nakapag-align na yung ating chromosome. So, ang very important lang na keyword na tatandaan natin sa metaphase is alignment. Alignment of the chromosomes at the metaphase plate or equatorial plate. Yan. So, they are perfectly aligned. Wala na tayong makikitang nuclear envelope. So, na-dissolve na or nag-disintegrate na. And then at anaphase, once alignment has already happened at metaphase, unti-unting mapu-pull. So, okay, yung chromatids ng ating chromosome ay, yung chromatids ng ating chromosome, this one, will be pulled towards opposite poles, papunta dito. And with that, nasa-separate na yung chromatids. Okay, we call this actually chromatids. But remember, one chromatid is equal to one chromosome. One chromatid is equal to one chromosome because it already contains yung ating centromere. So it's connected by the centromere. Centromere is already present. So in anaphase, one chromatid is equal to one chromosome. Ana means away. Away from where? Away from the equatorial plate or away from the center. Next. In telophase, telo meaning end. So, big sabihin, ito na yung last stage ng mitosis natin. Right? So, what happens at telophase is nagsistart ng ma-form ulit yung nuclear envelope. Ito siya. Okay? Diba during pooling or the separation of the chromatids, moving towards opposite poles, parang nagiging mas oblong yung cell. From this type, mas circle or mas rounded na cell at prophase and metaphase, kung nakikita nyo yung sa anaphase, ang itsura na ng cell is mas nagiging oblong siya. Mas nagiging oblong siya because of the pooling of the chromatids na nangyayari, because of the force exerted. So with that, that will initiate now yung tinatawag nating cytokinesis. Pag sinabi nating cytokinesis, this one, division. Kinesis is division, cytoplasm. So what happens is the cytoplasm will divide. So that at the end of mitosis or at the end of telophase, dapat nakaproduce na tayo ng 2 daughter cells. So madidivide ito. So kaya makikita ninyo itong parang nagsistart na ma-divide yung cell. Nagsistart na itong curve na that will represent yung cleavage furrow na tinatawag natin sa animal cell. And we call it cell plate kapag sa plant cell naman nangyari. So nag-reform, muling babalik yung nuclear membrane to protect the chromosomes of each daughter cell. Yan. And then, ito yung tinatawag ko kanina na cytokinesis, yung division of the cytoplasm. But, cytokinesis is not a part, or is not a stage rather, it's not a stage of mitosis because it's a process that happens during telophase. Okay? So, with the pooling of the chromatids or chromosomes towards opposite poles, unti-unti ding nagko-constrict yung microtubules. So, constricted remains of polar spindle microtubules, ito siya. Okay? Because nagkakaroon ng division of the cytoplasm. And then, finally, at the end of mitosis, fully formed na yung ating nuclear envelope protecting or surrounding the chromosome. Unraveling chromosome. So, single-stranded na yung ating chromosomes. Okay? Single-stranded yung chromosomes. Next, this is just a difference between cytokinesis in animal cell and plant cell, and it's just what I said earlier na cytokinesis sa animal cell is the creation of cleavage furrow. So merong naka-create na cleavage furrow using a ring of contractile protein, actin, and myosin. So basically, cytokinesis is madidivide lang yung parent cell para makaproduce siya ng 2 daughter cells. By the way, I forgot to emphasize, we have 3 chromosomes at the start of mitosis, 1, 2, and 3. And then, at the end of mitosis, still, meron pa rin tayong 3 chromosomes, each cell. Kaya sinabi kanina na the genetic and chromosome composition of a cell is faithfully reproduced in each of the daughter cells. So we have 3 chromosomes in each cell. Same as that with the number of chromosomes in the parent cell. So these are the consequences of mitosis. The chromosomes are reproduced and transmitted equally to the daughter cells. So 3 chromosomes at the start of mitosis, at the end of mitosis, 3 chromosomes then. Next, the gene retains its individuality regardless of the nature of its allele because the chromosomes hold the same relative position at late anaphase to telophase as they enter the next prophase in the succeeding mitosis, right? Another, the parts of the chromosome associated with the nucleolus remain associated during that interval. So kung napapansin niyo, naka-attach pa rin yung ating polar microtubule rather spindle fiber doon sa centromere. But if ever merong NOR or nucleus organizer region na na-form doon sa isa sa mga chromosomes, yung position na yon same pa rin until or hanggang sa susunod na phase or stage ng mitosis. Okay? And every cycle, doon pa rin yung nucleus. Hindi siya matatanggal. Nandun pa rin yung NOR or nucleus organizer region. Next, homologs that have mutated retain their separate difference mitosis after meiosis. Mitosis after mitosis. So, if may mutation man or errors na nangyari during the process, during the cell cycle, okay, during the events that happen from prophase, metaphase, anaphase, telophase, if there were mutations or errors, makakapi siya. May tutuloy until the next na stage or until the next na susunod na cell division na naman. So, yun. Yung mutation that happened, that's actually a process wherein may nag-change doon sa DNA sequence and that is actually copied from cell to cell. So, matatransfer siya. So, ibig sabihin, kung ano man yung mga changes or changes rather in the DNA sequence that will be that will become a source of variation. Okay? And then, meiosis, the chromosome number of the cells is reduced to half its usual number. Unlike mitosis, sa mitosis, again, yung starting cell natin, merong 3 chromosomes, say for example. And then at the end of mitosis, meron ulit 3 chromosomes. So, pareho lang. But sa meiosis, if we start with 3 chromosomes, sa end of meiosis, we start with, 3 pairs of chromosomes. Sa meiosis, we have 3 chromosomes only. Okay? So, 3 pairs, hindi siya pareho sa 3 chromosomes because 3 pairs is equal to 6 chromosomes. So, yun ang ibig sabihin sa meiosis. So, meiosis is yung process na pina-follow ng ating gametogenesis. Remember that gametogenesis is the process of sperm production or egg cell production, gametogenesis, production or formation of gametes. Yan. So, haploid actually yung napoproduce natin after or at the end of meiosis. So, to restore yung chromosome number, to restore yung diploid number at the start of meiosis, fertilization should happen because that restores the chromosome number of the species. All right. So, we have 2 stages actually for meiosis, meiosis 1 and meiosis 2. Yung meiosis 1, tinatawag nating reductional division. Okay. Reductional division because at the end of meiosis 1, na-half na yung number of chromosomes present in each cell. Sa meiosis 2, equational division siya kasi kung ilan yung chromosome number present at the start of prophase 2, ganun din yung chromosome number present at the end of telophase 2. Kaya siya tinawag na equational division. So for meiosis 1, we have prophase 1, of course, which has actually five stages or sub-stages. So sa prophase 1, what happens is that there are pairs of chromosome, which we now call homologous chromosomes. So ito siya, homologous chromosomes, because these chromosomes are already in pairs. Yung sa mitosis natin kanina, walang pairing na nangyari. Okay? So we have here 3 pairs. We have 1, this is 1 pair, this is 1 pair, and this is another pair. You might be wondering kung bakit 1 pair lang ito. This is only a 1 pair because kung titingnan ninyo, ito yung ating chiasma or since dalawa, chiasmata. Okay? So, yun siya. We have 3 pairs of chromosomes. Now, if you are wondering or if you are curious bakit 3 pairs, pwede ninyong gamitin itong previous slide natin. Okay? Because kung titingnan niyo sa metaphase, pag dumating na siya sa metaphase, 3 pairs lang din ng chromosome. We have 1, 2, and 3. Okay? Pero, since hindi pa siya arranged kasi dito sa prophase, hindi pa siya naka-align or hindi pa maganda yung formation ng mga chromosomes at prophase 1, then you just count kung ilan yung pairs na meron tayo. Okay? Since meron siyang label dito sa ating cell, we have here the chiasmata. So, ang chiasmata kasi ay yung point of contact or point of exchange during crossing over. So, ito yung ating centrosome. So, pareho lang actually yung mga events na nangyayari sa mitosis at saka meiosis. The only difference is sa meiosis, by pair. Especially sa meiosis 1, by pair siya. So, unti-unting pumupunta doon sa opposite poles yung ating centrosomes containing the centrioles, and slowly the spindle is formed containing the spindle fibers or the microtubules. All right. So, doon sa 5 stages of prophase 1, we have leptotin, zygotin, pachytin, diplotin, and diakinesis. The important things that you have to know in each substage is that in leptotin, the chromosomes condense. It is necessary that the chromosomes should condense so that they can properly align at metaphase. They can properly arrange themselves at the equatorial plate at metaphase. And they are still long, slender chromosomes at leptotin. Once they have finalized condensation, they will look like the mitotic chromosomes already, arranging in pairs. So that's it for leptotin. This is actually the nuclear membrane. Until the end of prophase 1, mako-complete yung dissolution ng nuclear membrane. Okay, so ang nangyari pa lang kasi at leptotin, condensation of the chromosomes or shortening, dapat ma-compact, densely packaged yung chromosome. Next, in the zygotin, the bivalence form. When we say bivalence, ito siya, the whole of it bivalent. Ibig sabihin, dalawang valence. Okay? Or a pair of homologous chromosome. Okay? Bivalence. So, naka-bipair na siya. Sa leptotin pa kasi, during condensation, during the start of condensation, hiwa-hiwalay or hindi pa maganda yung positioning or arrangement ng mga chromosomes. Sa zygotin, they are already in pairs. Yung pairing ng homologous chromosomes or yung formation of bivalence ay tinatawag nating synapsis. Synapsis means pairing. And as a result of the synapsis of the two homologous or of the pair of homologous chromosomes, what is formed is the synaptonymal complex. Para siyang magnet. Parang siya yung nagmamagnet nung pair of homologous chromosomes. Right? So, yung formation ng synaptonymal complex will now be leading. Parang siya yung mag-initiate ng crossing over. Okay? That is, if crossing over will happen. But even if crossing over will not happen, meron pa rin formation ng synaptonymal complex. Kasi nga, siya yung parang nagmamagnet nung dalawang members or two members of a pair of homologous chromosome. Okay? Because crossing over may or may not happen. Right? Okay, so pakitin na, crossing over happens, meron na tayong exchanging of segments. Pag sinabi natin crossing over, exchange of segments. So that, at diplotin, ganito na yung itsura. Notice that we have a blue chromosome here and a red chromosome as its pair. Pagkatapos ng crossing over, ganito na yung itsura ng segment ng chromosome. Yung blue na chromosome, meron ng portion of red, and yung red na chromosome, meron ng portion of blue. Because, or as a result of crossing over or the exchanging of segments. Meaning to say, the genes present from the blue are now exchanged with that of the red chromosome. Okay? And, yung crossing over, yung point of exchange nila, tinatawag natin kayasma. But if there are many points of crossing over, the plural is, of course, kayasmata. Okay? So ito na yung synaptonymal complex na parang nag-a-act as magnet doon sa pair of homologous chromosome will now dissolve or matatanggal na siya. Okay? Para during yung susunod na stages, mas mabilis or mas madaling mag-separate yung pair of homologous chromosomes. And then finally, at diakinesis, madidissolve na yung nuclear membrane so that as they enter metaphase, makakalabas na sila ng nucleus. Okay? Nakalabas na ng nucleus itong mga pairs of homologous chromosomes. So yun yung nangyayari at prophase 1. Okay? Metaphase 2, same lang din, alignment of chromosomes, but let's be specific, at metaphase 1, ang nag-align ay yung pairs of homologous chromosome. By pair yung nangyayari sa alignment. Sa mitosis kasi, iisang chromosome lang ang nandito sa equatorial plate or metaphase plate. Dito sa metaphase 1, by pair. Okay. Next, anaphase 1, separation. Sa mitosis, sa anaphase or sa mitotic anaphase, what happens is the separation of sister chromatids. Tama? Dito sa anaphase 1, sa meiosis, is what separates ay yung mga nag-pair. Yung mag-partner na chromosome kanina, sila yung magsa-separate at anaphase 1. Okay? So, ibig sabihin, the chromosomes at each pole that is being pulled still has 2 chromatids. Okay? Still has 2 sister chromatids. Next, telophase. Again, at the end of telophase, sabi ko kanina, madidivide yung cell as a result of cytokinesis. Each cell will now contain kung ilan yung chromosomes pulled at each pole. We have 1, 2, 3. 1, 2, 3, consisting of 2 chromatids each, still, ganun din yung number of chromosomes present on each cell. Okay? So, ayan, kapag completely divided ito, meron na tayong na-produce na 2 cells, 2 daughter cells from 1 cell at prophase 1. So, prophase 1, 3 pairs, right? 3 pairs of chromosomes or 3 pairs of homologous chromosomes. At the end of telophase 1, we only have 3 chromosomes na. Hindi natin siya tatawaging homologous chromosomes kasi yung term na homologous chromosomes ginagamit lang natin if they are in pairs. Okay, so from the 3 pairs, 6, na-reduce siya into 3. Okay, na-reduce siya by half. Okay, next, yung stages or the activities or the events that happen at meiosis 2 are just the same with the events that happen in mitosis. If you can see, kung marirecall ninyo, yung alignment ng chromosomes at metaphase 2 ay parang sa mitosis. The only difference now is that ang makikita ninyo sa structure or appearance ng chromosome ay meron ng genes or segments na iba yung genes contained. So meron ng, say for example, here, this red chromosome contains blue. This red chromosome also contains blue but on a different chromatid. This blue chromosome contains red portion. So these are a result now of crossing over. Okay, yun lang yung difference ng mitosis and meiosis 2. Okay, tinawag siyang equational division because the number of chromosomes in each cell is the same as the number of chromosomes at the end of meiosis 2. Right? So 3 yan. 3, and then we have here 6. We count 6 chromosomes here, because we count each chromatid being pulled at each pole we consider as 1. 1 chromatid is now considered as 1 chromosome at anaphase at anaphase 2. Okay? So yan, 3 chromosomes, 3 chromosomes. So from the one daughter cell or one parent cell at the start of meiosis, we now produce 4. So as a result of cytokinesis, this is divided. So completely divided. Yan, we will produce 2 cells, 2 cells. So one parent cell will produce 4 daughter cells. But haploid na siya. Haploid daughter or haploid set na siya. Okay? Because we have to produce haploid because gusto natin ma-restore yung diploid chromosome number via fertilization. So, the consequences of meiosis, na-conserve yung chromosome number. Ma'am, na-conserve yung chromosome number, pero na-half naman, naging haploid naman from diploid. The chromosome number is conserved if fertilization happens. Okay, so chromosome number is conserved from generation to generation. Yung namang mitosis, the chromosome number is conserved from cell to cell. So, the same from cell to cell. Okay? So each paternal and maternal chromosome has equal chance of being located at one or the other daughter nucleus. So it's either this came from the paternal cell, or it's either this come from the maternal cell. So the number of possible combinations of paternal and maternal chromosome is equal to 2 raised to n, where n is the number of chromosomes in the genome. And the probability of a gamete containing only the chromosome from one parent is equal to 1/2 raised to n. So if you are to look at, say for example, we have 46 chromosomes in each of our cells. Edi, we have 2 raised to 46. So there are many possible combinations. Okay, ibig sabihin, many chances of being not like us. Something like that. Or many chances of producing this type of genotype or type of phenotype and so on. So maraming chromosome combinations. Next, meiosis provides the physical basis of segregation of paired genes, which happens at anaphase 1. Because again, the separation or segregation of paired genes or paired homologous chromosomes happens at anaphase 1. Okay? Number 4, the number of non-sister chromatid exchanges, the number of different kinds of gametes with genetic combinations and recombinations is virtually infinite because of this probabilities and high number of possible chromosome combinations of paternal and maternal chromosomes. And this is very important because it is necessary for evolution to happen. Okay? Because the crossing over, the exchanges of segments between two non-sister chromatids cause variation. And variation or variabilities, genetic variabilities, contribute to the evolution, okay? Evolution of individuals. Lastly, life cycles. So this is just to show you the general pattern of eukaryotic life cycles. Although there are some variations of terminal orgametic meiosis, which happens in us humans, animals, intermediary or sporic meiosis, which happens in flowering plants, and then initial or zygotic meiosis, which happens in other species not mentioned. So ito yung general pattern, which is very important. So from a diploid generation, so yung diploid na cell or yung diploid generation undergoes meiosis. Okay? And then after which, as a result of meiosis, what happens is haploid. Mapuproduce. Ano yung mapuproduce from meiosis? From diploid, correct? To haploid na generation. And to restore yung diploid number, there should be fertilization. Okay? So yan siya. General pattern of eukaryotic life cycle. So very, very simple lang na discussion, just an overview of the general pattern of eukaryotic life cycles. So thank you very much for listening.